say, you have found a molecule. Okay, let's say this is active site. You got a molecule that binds to the active site pocket, right? This some kind of interaction. Let's say you have predicted to be having high binding affinity. Okay, so let's say -14 is your predicted binding affinity score, and when you did experimental study, you also get, like, if it is in the terms of IC50, so let's say you have 10 nanomolar something kind of thing, IC50. So it's very good, showing very good binding affinity, right? In vitro you have done. But is this enough? If you are going to do proper study, then we are not just looking for molecule that can bind at the active site pocket, but yeah, if you are doing the initial work, then you are looking for molecule that can bind to the active site pocket, and those are called as hits. Okay, and hit should not only have high binding affinity. Okay, it should be also be able to selectively bind to the target molecule. Why? Because it, it, it binds to the target molecule, but also binds to another molecule. Okay, I said it also binds to another enzyme or another protein. It can give side effect. Okay, so if you are having off target, then it can lead to side effects, right? So you should be looking for molecules which selectively binds your target protein. It should be able to absorb in your stomach, right? And it should be able to permeate to the gut walls or cells so that it can reach the target sites, and it should be metabolically stable. So when you take orally your molecule, supposed to be drug molecule, uh, should not be degraded by the liver enzyme. Okay, so it should be non-toxic, less toxic, right? And it should be synthesizable. Okay, so let me just show you one quick. So this is work that we have done few years back. Okay, so we are able to synthesize uh, a new molecule, okay, which is having very nice where is that? Yeah, I think this one. Yeah, so it's having 6.2. This is human acetylcholinesterase. Okay, so it is having 6.2 nanomolar. Okay, IC50 value, and donepezil drug molecule is having 8.2. It's better than donepezil. Okay, and we did what toxicity also. I think they have done toxicity also analysis. I think so. Yeah, somewhere it's there. So what was the important thing I want to tell you here is, so this drug molecule, though it is showing high binding affinity, I mean predicted as well as with the experimental binding affinity, okay, better than the drug molecule itself, but it is not able to cross the blood-brain barrier. So what is the point, right? We have synthesized the molecule, we got the binding affinity, high binding affinity, but if it is not able to reach the target sites where it should work, what is the point, right? So what we did was we tried to find molecule which can cross from this particular skeleton. We tried to find the scaffold, we tried to find the we try to design new molecule. Okay, so based on some method we are able to come up, but see this is a linker. Okay, so this single bond, one of the single, this double bond, if we turn this into single bond, it is showing high, you know, I mean, they are able to cross the blood-brain barrier, predicted. Okay, so they did. We tried to synthesize this new molecule without with a single bond here, but what happened is it is not, we are not able to synthesize this because some kind of condensation reaction happens when we remove this double bond and make it into single bond. It collapses. Okay, it's not, it is, we are not able to synthesize the molecule. Okay, even though it is having same predicted binding affinity as this particular molecule, and it is showing high, not high, but it is showing that it's able to cross the blood-brain barrier, but practically when we try to synthesize this molecule, we are not able to do it. Okay, practically we are not able to synthesize the molecule, right? So you have to look for, i'm just not I'm just talking, uh, if you are going for serious research, and if you are looking for more, advanced studies, then you have to look into all these properties. Okay, so these are ADMET properties, and also this that does not come under ADMET. The first and the second, the last one does not come under ADMET properties, but you should also consider these two uh, points. Okay, you should be selectively bind to the target protein, as well as it should be synthesizable. So if you are doing uh, serious research, then you should also be testing whatever you have predicted, synthesize it, and then test experimentally. Okay, okay. So when we say drug discovery, okay, we are looking for some of the steps involved in drug discovery, right? So the first step is target identification. The second is lead, identifying the lead, hits, then to lead, and then lead optimization. So most of the computational work that we can do is in these three steps, right? And again, drug discovery is a long process. It takes about 10 to 5 to 10 years, and it costs about $2 billion, right? So when you have identified the targets like we have done before, or nowadays you can identify novel targets also using genomic studies, right? So anyway, when you identify the target, the next thing is to identify the hit molecule.